All right. Good afternoon, all. I don't need this up anymore. All right, we'll get started in a few minutes. Just wanted to start the stream itself, just in case anyone had any questions for me before we get into translation. Again, this is short. We're not doing a ton with translation today. I do want to focus on the test itself, discussing what it's going to look like, what you need to do, and we'll go through the study guide. And I want to give you guys a chance to actually ask questions about the study guide and other things. So that's why we are going to be here. And again, because I know you're focusing on the test a lot, I want to make sure, again, if you've had the chance to look at the study guide, good. If you haven't yet, I strongly suggest you try to fill it out and answer the questions that are part of that. I'm willing to pull up any of the lectures that you need. So right now, I am swamped with a few things. So if you have turned in any of your if you've turned in any of the things that you were supposed to turn in, I haven't had a chance to grade any of them. As soon as I get the rest of the tests finished for this week, which there are several, I will be able to focus on the grading again, but it's been a lot. Trying to create the grades is a lot of formatting the exams because it has to be in a situation where it's accessible and it's good for an open book format, which is hard to format and figure out. And it works in the sense of, is it a fill in the blank question? Is it I could just write short answers, but it needs to be properly vetted so that all of you have a unique test. So there was no case study due this week. Okay. All right. Hold on. There was a case study. No, there's no case study due. I didn't post one, and it's because I wanted to go through we're going to do an activity today where I'm going to walk you through kind of all the pieces. We're going to put the pieces together in the case study period. Okay. I do think this will help you as well with this recent section. It's been a lot about DNA replication and the central dogma overall, trying to understand how that connects to genetics. So I think that this activity will help you to prep for the test as well. Okay. So we're going to do that together. You'll have a case study posted for next week by tomorrow, but there is no case study posted currently. Because we're going to have an activity today that's going to be graded, but it's a smaller activity because I didn't want to overwhelm you guys, and it's mostly to help you understand how to connect the dots together. Okay. All right. We'll get started in a few minutes. Hope you guys had a good Easter weekend. I anticipate that I know a lot of you guys are feeling stressed about the test because there is so much of that mapping on there. I will happily go through as much as I can during the second, between like 2:30 and 3:30. I'm happy to go through as much as we need to on the study guide. Try to help you answer the questions, help show you where I'm focused, etc. Okay. Fortunately, it does look like, though I know a lot of universities have contingency plans, it does look like things will hopefully be back to normal over the fall. If you're planning on taking summer classes, it will not be back to normal yet. summer will be the time when things are kind of phased back in. So we are still having online classes this summer. There will be normal classes the semester after. There. Okay. So we're gonna get started pretty soon. Hello to anyone who's just coming in. I know that Faye is here. I don't know who else is here. Again, for today, we will be working on we'll be going through just the translation. I'm going to be putting it all together. Hello, how's it going? And we're going to be talking about the case study that we're doing. It's more of an activity to discuss how the central dogma that we've been focusing on the past few weeks can lead into understanding genetic diseases better. Okay, I think that's a useful one to do. All right. So again, we're just going to finish up translation, but we're going to do that pretty quick. Translation, I'm not focusing on us heavily. We're not looking at protein structure and how proteins can refold and all the different functional groups. That's biochemistry. That's a different thing, okay? You learned a lot about the proteins already. I'm not going to bore you with a lot of those pieces there. I wanted to focus on mainly how the flow of information can change and gene expression can change in terms of genetics, okay? We're going to start jumping into gene mutations and then what that means for the body on Thursday, and then move into the talking about genetic recombination in more detail. All right. So we talked about the degeneracy of the code already. Each amino acid may be specified by more than one codon. Synonymous codons can specify the same amino acid. In transfer RNA overall, it brings the correct amino acid to the ribosome. Its anticodon is complementary, has complementary base pairs with the codon of mRNA. Again, this is Bio 1140, right? And each type of tRNA attaches to only a single type of amino acid, okay? So this is the basic gene flow overall, okay? DNA has this information that goes to the codons in the mRNA. And mRNA is the code. DNA is copied onto that, but DNA is the opposite of the code, okay? And the anticodons here are the same as the DNA molecule, except for the fact that there's U's and some of those pairings are wobbly. Okay, that leads to the actual code of the amino acid. Again, this wobble hypothesis is that the pairing of the third codon is relaxed. This allows one tRNA to wobble and attach the same one at a few different ones, and this is because the first two are more important to the identity of the amino acid than the last one. Last one can wobble a little bit. It can pair with C or with U. A purine cannot bond with a purine, but it can bond with either of the other pyrimidines. Okay. Again, this is a flexible pairing in the third position. This again allows for there not to have to be so many tRNAs created to match every single one of those, but just enough to get the right amino acids. And again, this has to do with the purine pyrimidine pairing. And the way the degeneracy of the code works is that generally, CUC and CUU, the pyrimidine or purine order can be reversed on there, and the last one is wobbly. The other two are not. And if you look at the way the code is set out, you can see that matters in terms of pyrimidine versus purine, matching the size, and that's only in the third one. Okay. All right. So again, we want to understand that normal base pairings and wobble rules. Okay, so here's the wobble rules. Understand that if it's G, it can bond with C or U. C can still bond only with G. A can bond only with U, and U can bond only with A or G. So with A or G, the concept here being that, again, there's some wiggle, but the wiggle is small, okay? Generally U and G are the ones. C can still only bond with G, A can still only bond with U, but U can wobble around with A or G, G can wobble around with C or U, okay? So if it's GUG, if it was just the straight pairing, that would be CAC, right? Are there any others that it can bond with, that it could pair with if you go along with the wobble rules? So it can also, Beatrice thinks it can also bond with B. And so I guess the question would be, can it bond with C? Okay, so D would be all of the above, but note that C is going to only ever bond with G, plus the wobble only happens on the last one, okay? So G can bond with C or U. So this is a wobble rule, A and B. But it cannot bond with UAC because the wobble is not on the first codon. It is on the last two. It's on the last one, okay? So this first codon is different than the actual tRNA anticodon, and the tRNA itself will be different, carrying a different amino acid because the first two matter, last one less, okay? So it's going to be E, both A and B. So B can also bond. So if you said B, you're correct. It cannot bond with C, again, because the wobble rules only have to do with the final one on the 3 end, not on the first one. The first one can't be different, okay? G must bond with C on the first one. U must bond with A in the second one. The third one has the wobble. Okay. All right. Genetic code is also non-overlapping. We covered this a little bit before, but the translation reading frame is usually set by an initiation codon. Okay, remember that the mRNA that comes out also contains the promoter. The promoter generally is not AUG, but the actual start of where the gene is marked is generally marked by AUG. Some other locations can be marked as start codons, but they're rarer. But usually AUG is the start. Okay, so the non-template DNA, okay, the one that's not red, we'll say ATG, and then the AUG will be the mRNA because it looks like the non-template. Again, the template DNA is going to be the opposite there. TAC, AUG. Again, you need that reading frame to be correct. If insertion errors, again, we'll talk more about genetic mutation and such, but insertion errors can be the most dangerous in the sense that it changes the entire reading frame and could then change the entire pattern of amino acids, which is why something like the one in the muscular dystrophy that you guys have been writing about, that muscular dystrophy one is more related to the trouble in the reading frame than it is to that single amino acid being broken. The key here is that it's the reading frame shift that really causes the big issues. We talked a little bit about those areas of mutation. The code is nearly universal. Same codons specify the same amino acids in all organisms, which does also mean that we can take, this is why viruses are able to use RNA reverse transcriptase to basically use our machinery to build its own RNA, because we have all the materials to build its RNA and we can read its RNA. So it's an interesting pattern there. But that's why viruses exist the way they do, because it can build itself using other cells' ability. All right. So here you have all the pieces. Okay, so the ribosomal unit has a large part and a small part. We didn't really go over what a ribosome is. It's just a protein plus some rRNA. Remember, that's read by a different RNA polymerase. Translation, like transcription, 3 phases. We've got initiation, elongation, and termination. One of the key things I want to focus on is the termination part, because that really is where a lot of the information about where to stop is. You can have a long mRNA, but it has the stop codon in the middle, then it stops. And that's really the important piece to understand. Okay, so again, you've got this untranslated region. That's the region of the mRNA before the start codon. It does not attach to this part. If the AUG was over here, this 5' untranslated region would be a little larger. It always starts at the 5' and reads to the 3' end, okay? It does not get to the poly A tail unless the stop codon was somehow altered, and then it would have a much longer protein than it needed, okay? And it could read that poly A tail, but generally it's going to stop before that, because there will be a stop codon in the actual protein coding. Protein coding area is flanked by the start codon and the stop codon for a perfectly working gene. And we'll talk more about gene mutations later and how that can impact some of the actual protein translations. But okay, so one of the keys here also is that this poly A tail will kind of thwack over onto this ribosome, and it holds it in place, so it's not just like wobbling, wiggling, and breaking over here. This helps stabilize it, okay? So that poly A tail has some benefits. Okay, so you got the ribosome, it's attached, and then it starts reading at AUG. It doesn't start reading until later, so it attaches first, finds that first one, goes down from 5 to 3, finds the AUG, and then starts building. All right, so you've seen this before, right? There's basically 2 stations in each of well, 3 stations, sort of. The concept here is that you have the addition of, so one of them plugs in, one of the tRNAs plugs in, it attaches, it finds the matching codon to its anticodon, and it's holding on to the correct amino acid for that codon, and then it gets pushed out by the next one in the line, and as it's pushed out, these bonds are formed, okay? We're not going to worry about bond forming right now. That was all taught, the actual how do you form these polypeptide chains. That was all covered in 1140, and not as important to what I'm trying to focus on here. Really, it's is the transcription and translation working properly? And so understanding that piece, okay? So if it says build AUG, GGA, UGU, okay? That's going to, first you're going to have the methionine, then glycine, then cysteine, okay? And those match with these amino acids. Again, there's a little bit of wobble here, but generally those wobbles are not. And as these come in and it moves down, it pushes these off and it forms the polypeptide bond. Termination is one of the things I wanted to talk about. The ribosome proceeds along the mRNA and one codon at a time until a stop codon is reached. No matter when this is reached, that's the end of the translation. It's not going to go back and retranslate it, okay? And note that the mRNA, after it is read, degrades afterwards. You don't have it read multiple times by multiple ribosomes. This is done. The end. So you need, and there's probably multiple mRNA molecules out there that are being read, but this is the key, is that if you have a stop codon, that's the stop. And again, it's downstream of the start codon, downstream being in the 5 to 3 direction, okay? UAA is a stop codon. Instead of attracting one of the tRNAs, like this one here that's the anticodon of GCU, okay, and attaches an arginine, it attaches this release factor. This release factor, what it does is it puts on a water molecule instead for an OH group at the end, and it signals these two to release from the mRNA. Okay, so what the reason I wanted to focus on this a little bit was that the termination piece, one of the key things is that when there's an error in the, so there's nonsense errors. When an error is occurring in the DNA, it reflects on the mRNA. And if the error in the DNA causes the mRNA to read a stop codon, instead of where there should be a protein, you can have missing proteins. The missing proteins are things like where there should have been melanin formed, there is no melanin. So it breaks, it's not a functional melanin, and you have albinism. Or it's a precursor to melanin, as we had talked about before. That precursor protein to melanin. So stop codons and nonsense mutations are some of the more intense of the gene mutations because it can cause entire proteins that are vital to disappear. Also, you know, even just having a single missense where you change one codon into another, that codon change can change the shape and thereby the functioning of the protein, because shape is very important to protein participation here. Okay, so do a brief review. Through wobble, single blank, compare with more than one blank. What's pairing with what, I guess, is the question here. Now, note that the tRNAs, so we have a D here, the tRNAs are not destroyed after this. mRNA is. It's a very unstable molecule. That's why you have the caps on it. Remember that tRNA is not destroyed. Okay, so you have a lot of Ds, and that's true. A single anticodon can pair with more than one codon. The codon remains still, but the concept here is that if you have like 50 tRNAs out there, that one tRNA with the GCU can also pair with, it can also put a different one at the end, okay? So that wobble, and you can have that tRNA working over and over, bringing more and more amino acids from the cytoplasm to there, okay? Any given RNA sequence has blank possible reading frames, and the correct one is set by a, because I'm not fully, so I guess I didn't cover the number of reading frames it could have, but it's always a correct reading frame. So, sorry, I should have checked this beforehand. I'm not sure why it's 3 actually, so don't worry, you won't be tested on that. So it is marked by a it is marked by an initiation codon. I didn't check this question ahead of time. I didn't teach you about the reading frames that RNA could have. So don't stress out about this one. I'm not testing you on that. I don't remember why it's 3 reading frames. So initiation codon was really what I was going for. All right. Again, as I said, I'm not spending too much time here. This is a pretty simple piece. And we're not talking about protein structure yet. I will talk about that a little bit more as we get into gene mutation and how that can impact functioning of organisms. But this is the last piece. When we're talking about how gene expression can change, well, translation doesn't change much. If there's an mRNA to be read, then it'll be translated. Not very many things can mark an mRNA to not be read after it's in the cytoplasm. There's a lot of pieces that can wobble around all of the where is it spliced and how do the exons go together and which intron is spliced out and how does the and all sorts of things that happen in transcription, the mRNA processing, the actual coiling of the DNA, the actual access of RNA transcriptase, the way that different things are replicated. But translation is a little bit low on the information. It's just following the plans that were set out. Changing the plans is easier than actually changing them being set out, is the concept here. Okay? All right. So what I'd like to do for the rest of the time here is actually let's go through let's walk through a little bit of the let's walk through the study guide. Okay. Save that. So the study guide really, it's here. I'm gonna make it bigger, and we'll just walk through some of the pieces. I know that some of you guys were worried about different parts. So note that a lot of what I grade on is more concepts. Okay, I don't just need you to be able to write the exact chromosome definition, okay? So you need to know what a chromosome is. But mostly a lot of these pieces are not just, you're not just going to be doing define the vocabulary word. The key here is understanding what I mean when I say linked genes and then being able to solve the problem about when a linked gene is happening. Being able to understand what I mean when I say 3-point test cross so you can solve a 3-point test cross problem. What do I mean by the trans versus cis orientation, and how do you solve a problem that has a trans versus cis orientation in a linked gene piece, okay? So a lot of these are not going to be just defining. If you know what they are, you need to be able to know how to use them, is the concept. So for all of these, again, this is more like be able to calculate the coefficient of coincidence. Not just what is it, but how do you calculate it? Not just what is interference, but how do you calculate it? Not just what is deletion mapping, but how do you read a deletion map? Okay? All right. So if you guys have had a chance to look at this, then you can ask specific questions. Otherwise, I can just run through them. And but if there's anything specific you want me to focus on in here, we could do that, okay? Again, for chapter 7, a lot of these I'm not going to, in the questions I'm asking you in the test, I'm not going to ask you to define what parental means. What I want you to be able to do is if I give you a question and it talks about parental versus recombinant, I want you to be able to understand what I'm talking about, okay? All right. So the basics are all here again. You should know why these break the rules of Mendelian inheritance. What does Mendelian inheritance normally have? That independent assortment, why don't they have it? what the behavior of incomplete, completely linked, and unlinked genes look like. And then, maybe we'll go through a couple of these questions. Why are genes that are very far apart more than 50 map units hard to distinguish from those on different chromosomes in terms of when you do a breeding activity on them? Does anyone remember? Okay, well, we should cover that. They act like unlinked genes in the sense, note, in terms of frequency, they act like unlinked genes, okay? Because if it's more than 50%, then let's type this in here. Okay. So they act like unlinked genes. I'm going to type in the chat briefly. The concept is that normally, normal unlinked in a 2-gene cross, the gametes will come out, A, B. So this is for a test. If you do a test cross, okay, is that you're going to come out with for a heterozygote, okay? So you have a B, A, B, A, B, and B, okay? So in a 2-gene cross, you normally have 4 gametes. And in terms of those gametes, those are all in equal numbers. If you have a genes that are very far apart, then they're going to act unlinked. And the reason they're going to act unlinked is because there's so much space in between them, it's likely that they will cross over. Again, sometimes you can only tell that they are linked to a specific chromosome because they're, say, inherited with the X or something similar to that. 2 or 3 are inherited with the X. But if they're far enough apart, they're still going to act like it. Because if you have more than 50 units, you have more than, you have about 50% recombination. If you have more than 50 units, and even in an AB over AB times, you're still going to get in equal parts. Okay? Again, these can still be linked, but you can't tell in a breeding one if they're too far apart. So because 50% recombination, once you get far enough apart, it's going to just be equal there in the other parts. Okay, so that crossover will be as equal as it is in any other part of the chromosome, indicating that now you can, that recombination will be very common, and it looks like, but it's not more common than any other recombination, okay? So once you get above 50, you can't tell just by that, because it would be a 25, 25, 25, 25 pattern for all of those 4 gametes. Okay. So now we have, how does linkage impact inheritance? You should be able to answer that question. Obviously, we've talked about how it's not this 9 to 3 to 3 to 1 pattern in a 2-gene. Let's see. Understand what's causing it, the frequency of linkage breaking, how you write the notation and understanding notation again. The cis versus trans, okay, so this is a cis orientation. This is a trans orientation. That just means in a heterozygote, are they both on one side, or are the 2 dominance on opposite sister chromatids. That's the key. And then you have the output being different. Okay. Again, you should be able to calculate the distance. So in a specific recombination, in a 2-gene inheritance problem, you should be able to figure out the recombinant frequency and the number of individuals that you'd expect to be recombinant. Percent of total recombination is unit of distance. Remember that you have to have both recombinants, not just one of them, to figure it out. If I'm asking about a specific phenotype or genotype, then you have to answer with the frequency for that one and not for all recombinants. So just pay attention to what I'm asking in the questions, okay? All right. And again, a double crossover between 2 linked genes produces only non-recombinant chromosomes, since the second crossover reverses the effect of the first. This is why, if you want to find out, if you want to actually see double crossovers, you have to look at 3 genes, because the double crossover will change the location of 1. Okay. So I'm going to put up these pieces here, okay? Does anyone have questions about these recall concepts, okay? What I mean by any of these. If you'd like me to expand on one or more of them, I can. Which part? You're good on them. So I'm assuming that, Rana, you're good on them is what you're answering. You're happy, you want me to move on. Okay, for the true breeding. True breeding, I'll type it in here because you guys can see this too, okay? True breeding would mean something like ABB or ABB, okay? That's what true breeding is. So if I say something is true breeding, and I say it's true breeding for all recessive, then I mean it's homozygous recessive for every single gene that I mentioned. It can only produce when crossed, and so this is why we normally use this. This is what's used for the test cross, because it can only produce recessives. So we know which recessives came from the test cross parent. We're trying to find out which recessives or dominant came from the not test cross parent. The parent of interest that we're trying to look at the distance between linked genes. Anyone else here? I know there's a small delay. Okay. Sure. Let's talk through an example of mapping. Okay. So again, this you should be able to create a 3-point cross map sort of thing. Okay. So in terms of that, the pattern of problem solving is in the chapter 7 part, but the concept here is that if you have 3 linked genes, okay. So 3 linked genes, let's say A, B, C, okay, or I guess it'd be A, B, C. And you have a heterozygote. Let's say ABC, ABC. You don't even have to know though, okay? Because you can actually tell me from the information what's the parental genotypes or phenotypes. What are the parental phenotypes, and what are the double crossovers and what are the single crossovers? Okay. All right. We can go through more of the math, but let me just walk through the basics of how you build it. Okay. So I give you information about so heterozygote for 3 linked genes is crossed with okay, that's the true breeding recessive. Okay. The data for offspring will tell you parental most, double crossover least. Okay, so the others are single crossovers. Other phenotypes are single crossovers. The one gene different from parental is the middle one in the double crossovers. Okay? So if I showed you something where parental most common is AABBCC. Sorry. What I would tell you is that they are, when they express something, so I'm not going to write down the entire thing. You just assume that on one side is this at all times, okay? Because it's coming from this individual, right? But I'm going to tell you, you know, it looks like this. Okay? And and then this one looks like this. A, B, C. These two are the most common, okay? What it indicates is the orientation of their genes, okay? So can anyone type out in this formatting then what the parental, what does their genotype look like in terms of what's on what side? What does the actual parental look like? The one that's heterozygote for 3 linked genes. What is that parent? How are the genes oriented? So, well, the heterozygote, you're close here, but what I'm actually looking for, these we know both came from the heterozygote, both of them, okay? Because these, all 3, had to come from the other one. So at least half of this always comes from this, okay? Sorry, I wrote this wrong. It should actually look like this. So actually, that means that hetero parent equals this. Again, it's a heterozygote, But these are the two, these are its two homologs, okay? Homolog 1, big A, little b, big C. Homolog 2, little a, big B, little c. Which still equals, in genotype, but that's the concept, okay? It's still a heterozygote, but this is the orientation of its genes. So this is how it's going to come out. In the most common, each of them have one side that's coming from the parent and one coming from, so we're seeing the expression through the phenotype. So if they have a recessive trait, this one I would describe as recessive for A and C. This one I would describe as recessive for B. Wild type for the others, okay? These individuals, okay? So this would be like, 240, and that would be like 270. They're going to be about the same pattern, same frequency, okay? All right, so now we figured that out. If I told you that the double crossover, the least common was, let me look, which one can I change around? So I told you that the least common was looked like these two. Do I have the order correct when I type out ABC in the way I'm currently typing it? Is this the actual order of how they are arranged in the chromosome? No. How are they actually arranged? What is the actual order? Just type out 1A, 1B, and 1C in the correct order. What would be the correct order? Look at the ones I highlighted here in the parental. What changed? What's different here than the parental? Which one moved? Which one flipped sides? The C. C is the one in the middle. So your actual order is A, C, B. Okay. So you can make a map from that. Would be, so you have A over here, some distance, C, some distance, B. To figure out how the actual numbers that are corresponding between A and C and B. Now we have to organize the others, okay? So I'm not going to give you all of the, do look at chapter 7, I can pull it up, but the concept is now you have information about which of these are single crossovers and which of these are double crossovers. And the double crossover looks different than the single crossover, but now you have to, in order to get distances by adding single crossover in A to C region plus, so you're going to see the single crossover in the A to C region is going to look like this. Single crossover in the A to C region remember it's ACB. So if it's happening here, the A is going to switch places. So from the parental, so that's going to be an A, B, C, ABC, and A, B, C. Okay? So you add those together, plus double crossover, and then total crossovers divided by total offspring to get frequency and distance. Do the same with the C and B, okay? When it happens in between the C and the B, you're going to flip B, okay? So B would be flipped, so it would be big A, big B, big C, little a, little b, little c. That would be the single crossover in the C to B, okay? Are you following me here? There you go. So now you have those information. So now you have a frequency, which should be some sort of decimal, and a frequency, which should be some sort of decimal. Okay, so you have the decimal for both. You can express them in map units, but let's leave them decimals for now. Let's say you find out that A to C equals, let's say, 10%, and C to B equals, so that's how many crossovers happening. So this should be, your frequency is 0.1, 0.1. C to B is 0.23, okay? Concept. All right, A to C, 0.1, C to B, 0.23, okay? To get your, and then you get your expected double crossovers is going to be 0.023. It's a tenth of that. You just move the decimal over 1. There you go. Often, though, the expected double crossovers are not the same. First, you get the coefficient of coincidence. Okay, coefficient of coincidence. Who remembers how to calculate that? Again, you can look that up. I always forget whether to put the, in the coefficient of coincidence, do I put the expected on the top or the observed? I think you put the observed on the top over the expected because expected is usually bigger. That makes more sense. Okay, so now you have expected double crossovers, but then you put, so coefficient of coincidence equals observed over expected crossover frequency. So let's say you saw, we observe 0.12. Sorry, not 0.12, 0.12. Should be less. It can never be more, okay? But if we observe less, then there is interference. So if we observe 0.012, You put that over the template 0.012 divided by 0.023, and that equals some number I've forgotten. Okay, so divided by 0.023. Okay, that's 52. Okay, 52%. 52% of the point actual expected crossovers are happening. 48% interference. So coefficient of coincidence is basically the percentage of the expected crossovers that are actually happening, and that minus so 1 minus that equals the amount of interference. Okay? Did that help the whole pattern here? Again, I will post this current stream we're doing, and you can look at what I've written out. I could also, good, I'm glad that it did help. I can also copy paste this whole thing here. See if it goes in the chat, okay? Probably not. Nope, doesn't like me. Never mind. If you'd like, I can post this in the study guide area kind of thing. What we actually work through, that's fine. Gives you a pattern for what to do. Okay. I will post it though, okay? So you have a pattern of how to do it. Again, none of these numbers are gonna line up, okay? You still have to do the whole process, but this gives you a map of how to start building the pieces together. How do you actually put it all together? How do you actually put the pieces together that need to be together, okay? All right. Let's move on, I think, so that you guys have an idea of what to do for the rest of the pieces. All right. Bacterial recombination. I didn't really put that much in here. Because this really wasn't my big focus, okay? I introduced bacterial recombination because I wanted you guys to start thinking about how recombination and the interesting kind of genetic pieces that have to go with it. But mostly, you know, understand that there's plasmids and chromosomes. If chromosomes have the vital stuff, it picks up random plasmids. Transfer information and how, conjugation, transformation, transduction. Mostly we talked about conjugation and the movement of plasmid information from one to the other. Also transformation, it can just pick it up from the surroundings. And you likely did this in 1140. Be able to identify a vertical versus horizontal gene transfer. Understanding how the bacteria and the sea slugs can work together to move you know, those genes from the sea slug into a gene from the plant. A gene from the plant into the sea slug. Likely due to bacteria picking up and dropping off little snippets of information. Again, don't stress too much about this. The most of this information was how do bacteria move data around. How can you use bacteria to become factories? This is the basics of bacterial recombinance. I didn't focus on the rest of those pieces as much, but the vertical versus horizontal we talked about a bunch. Okay? Again, maybe 2 questions total on the exam about this one, because it was very short and I decided to move it more towards the end when we actually talk about GMOs and genetic engineering, because this is more related to that. Okay, so let's move on to chapter 9. All right. In this one, I have a bunch of questions. If you answer about the questions and understand really which one's which, remember the pyrimidines are the small ones, purines the big ones, which is a purine, which is a pyrimidine, understand what the differences are between them, and kind of how that all built into the central dogma, know the Chargaff's rules, complementary base pairing, deoxyribose versus ribose, what's on either end, that 5' phosphate unreactive, 3' hydroxyl reactive. And then, yeah, this should be mostly a review for you guys. I just wanted to identify, just have you understand a little bit about, in terms of DNA analysis, how to understand, there's these hydrogen bonds, and that relates to the difficulty of separating the sequences. Remember, G and C have 3 bonds together, making them a little bit more difficult to divide, and the DNA melting and denaturation point is at a different temperature than ones that have mostly A and T. Okay. All right. Again, remember all the Chargaff's rules. Be able to tell me if I gave you some information about the levels of a certain base, can you tell the other bases from it and how? Okay. And what the ease or difficulty of pulling apart things would be. What is information about the frequencies tell you about single-stranded versus double-stranded versus RNA versus DNA? So gives you some information. If someone gave you data about a DNA, about a piece of genetic material, you'd be able to tell me, is it RNA, DNA? Is it single-stranded, double-stranded? And then some information about how hard or easy it would be to pull apart. Okay? Again, a lot of review, but those are the basic pieces because it has to do with genetic analysis and kind of how DNA is structured. Anyone have questions on chapter 9? Confusions about it? Remember, chapter 10 was kind of linked to this because we talked about replication there. So replication, there's going to be a little bit more covered in terms of the process there. Understanding how the process is related to of DNA division is related to the structure of DNA and how it actually builds a new DNA molecule. But this is just RNA and DNA structure, which again should be mostly a review. Good? Okay, this one has a little bit less calculations in it, mostly. Again, maybe some of the ratios. Making sure you can do Chargaff's rules properly and that kind of thing. But again, not as much calculation as some of the previous ones. DNA replication. Okay, remember you had all these different kinds of replication. Make sure you go through that table that's in the chapter 10 guide. What's the difference between theta, rolling circle, and linear eukaryotic? Okay. Those different forms of DNA replication and why they do what they do. Okay, so just some information about that. What it means to be semi-conservative, the direction of DNA replication, bidirectional versus continuous replication, all of these different pieces, the Okazaki fragments, telomerase, telomeres, topoisomerase, DNA helicase, primase, the placing of the RNA primer, single-stranded binding proteins. This one, it was in the information. I didn't go over it as heavily. Just know that DNA polymerase 3 is the one that elongates the chain. DNA polymerase 1 is involved in replacing the replacing the primers in the lagging strand. It replaces them with DNA. Those are the two activities of we didn't cover all the various DNA polymerases. There's a bunch of other ones, but those are the two I want you to know, okay? That they just do two different things. The main activity is 3. The replacement of the chunks is this, and then ligase. This is not on here, but it should be there. Anyways, all the different parts that build it, be able to explain what each is doing and why. Okay. It's a good question. What about 2? 2, I believe, is one of the proofreaders, but it's not actually a little bit less involved in DNA replication itself. It does polymerase, but it's not. So there's more activities in 4 and 2 that are unrelated to the main progress of actually replicating. I don't know why it's 3 and 2 and not 1 and 2. That's weird. Why is it not 1? Usually these refer to the order of discovery. Okay, you won't be tested on 2, you won't be tested on 4. It does go through them in your book, but I'm mostly looking at the ones that are actively building it during the period of DNA synthesis. And I recommend, in terms of this, I posted a couple of videos that kind of outline this a little bit better. Just make sure that you are familiar with kind of the structure of the whole, what's the hollow enzyme, which kind of structures and divides the entirety of the linear eukaryotic chromosome. I only focused on a little bit of what's different between eukaryotic and non-eukaryotic, but all of these same the same enzymes are part of that. But again, please make sure you know what's doing what and where and how. Okay, so those pieces are going to be important to understand, because understanding the process of replication and how it is replicated perfectly is important to understanding how we don't have constant errors, basically. All right. Again, be able to explain what the end replication problem is, and then also how telomerase addresses it. I don't think I actually talked about that one, so you can just ignore that piece, because we didn't talk about that in particular. We were just mainly talking about the main process of replicating DNA and, the templates and having either side code for both sides. All right. Any questions here for Chapter 10? Again, it's just the basic replication, but do understand the replication is different between different organisms, and that those patterns are different, where the replication bubbles form, etc. Okay, Okazaki fragments. Okay, I'm ready to move on if you guys are. Let's move forward. All right, now we break out a little bit into the new stuff that you didn't cover as heavily, because we're getting into gene expression, okay? Chromosome, euchromatin, heterochromatin, the different kinds of supercoiling. Remember, the negative supercoiling opens the chain, positive supercoiling closes it, okay? And then what the histones do, how various activities impact availability of histone activity, epigenetic changes that you can turn on and off different genes via chromatin restructuring. So that's one of the key things I've been focusing on here. How are euchromatin and heterochromatin different? How is chromatin related to epigenetic changes? Positive versus negative supercoiled DNA and how that is different. Histone tails are positively charged. Now, the deal here is that I wanted to make this a little bit clearer, is that acetylation in general. Now, I know there's a lot of variation and depending on where this is all happening, but overall, acetylation means it means that it has neutralized the positive. Remember that DNA is negatively charged. Histone tails are positively charged. Acetylizing the histones, acetylizing the DNA means that you negate the positive charge. You let go of the DNA, which allows it to be read even in heterochromatin. Methylation does the opposite and wraps it tighter, okay? It encourages the histone tails to close and wrap tightly around the DNA, turning off the accessibility of its genes, okay? Those are the key things I'd like you to know in that sense. Remember, the constitutive being the parts of the heterochromatin is the parts that are near the centromere and the telomere. The facultative is the stuff that can be tightly coiled or not, depending on gene expression. So just go back and look through that, and you can make sure you understand the difference between those two and can explain what's happening there. All right. How you guys feeling about chapter 11 and understanding the relationship of chromatin and chromosome structure to DNA transcription? Okay-ish? Okay. You know, read through it, make sure you're comfortable with it. Look back, kind of understand those pieces. Again, understanding what happens. So, in this sense, understand the most accessible DNA is in euchromatin. DNA strands negatively supercoiled. Okay? If they are methylated, if it's in heterochromatin, if it's positively supercoiled, all of those pieces can make DNA not accessible to being read by RNA, being transcribed by RNA. Okay? Anyone else have questions specifically? All right, I'm moving on. Again, transcription and RNA processing was mostly what we went through. The spliceosome, the transcription unit, the genetic code and wobble hypothesis we went through briefly. both at the beginning and the end of chapter 12. The different RNAs, transcription units, what it means by upstream and downstream. Remember, it builds itself 5 to 3. Okay, upstream would mean upstream of the building point, and then downstream is the same direction as the RNA transcription is occurring. Okay. The termination patterns. Okay, be able to identify what's happening in each one of these. The Rho dependent versus independent. Remember, Rho is a protein that signals termination versus independent being a shape. It induces this cloverleaf. Exons, introns, protein coding read-in, 5-cap, poly-A tail, RNA splicing, alternative RNA splicing, exon skipping. We've covered all of that. And this was one of the key things I was trying to emphasize here is that RNA access to DNA, RNA polymerase access to DNA, the process of identifying locations to do transcription, the sigma factor, the ribosome, the different sigmas that recognize things for rRNA and tRNA, the messenger RNA and the TATA box, and all those pieces. Okay, so again, this is a little bit more complex here because we're not just talking about the process of how does it transcribe. The key here is we're talking about how does it turn on, how does it turn off. How can it be spliced variably to create variations on proteins? How is the RNA edited after? Why is it edited? How it knows where to start and stop? How it can be told to not start? How it can be told to stop? Okay? And all these pieces here, okay? Functions of these guys. What I recommend you do, again, some of this is new information, some of it's not, okay? I'm sure you learned about the degenerate and unambiguous genetic code already. But things like the process of transcription, in what direction does it move, how does the template and non-template DNA compare to the RNA transcript, again, the core RNA polymerase, sigma factor, this hollow enzyme, additionally, those outside activators or silencers, okay? So understanding all those information. What does the promoter provide to RNA polymerase? I suggest you go through, before you take the test, just spend a few minutes and say, can I answer this? How would I answer this? Can I answer that? How could I answer that? Okay? How do these different parts work? How do you process eukaryotic pre-mRNA to mRNA? What is the function of doing these various things? We didn't talk much about RNA editing, but that's another way that your body can actually change the protein that comes out depending on the actual environmental needs of the body. Okay. Alternative, traditional. The end result of alternative. Okay. All right. In terms of transcription and RNA processing, again, some of it's memorization. Some of it is just going back through and watching a couple videos and understanding what's happening during transcription. How do all of these pieces work together? And in particular, which we're going to be covering today during our period. Again, so in about 15 minutes, we will move towards actually doing a case study. Our case study today is really just a discussion, and we're going to go through a little game. So I'm going to link the game. The game is actually already linked. But the game itself, it's going to be part of your assignment. If you can't play it, you don't have the proper stuff, don't worry about it. The key here is that this game just gives you examples about different pieces. But one of the key things in transcription here, I wanted to heavily focus on is how, in terms of genetic engineering and medical technology and all the stuff we've been learning about, how can we change our own expression of genes in terms of disease management? A lot of these pieces have to do with that, okay? What if we changed where the promoter was going to be, added more activators, silenced a gene, caused those exon cutting drugs to occur? The protein coding regions, alternative splicing, changing the splicing, changing the chromatin structure, changing how, again, once it's out of the nucleus, this mRNA is sort of done and ready to go, whatever it is. But how can we change things in the nucleus to impact how mRNA is read? So those are the pieces I really want you to get out of this, okay? That's the genetics part. There's a lot of cell biology piece in here. Key here, how does it recognize a gene? And how does it interpret that gene? And how does it tell where to start and stop? How is that interpreted and changed and spliced and edited? And why is it changed and spliced and edited? So all those pieces. Questions about transcription. We're going to go through this again in a few minutes. We'll stop soon. But those are the basic ideas. Okay. If you have a specific question right now about transcription or RNA processing, please ask it. Otherwise, we're going to go through some fun activities here and talk about where are the points at which human gene editing can impact gene expression. Okay. I don't hear any more, or see, can't hear you guys. I don't see any more information from you guys concerning all of this, okay? So, let's go for we're good. Again, these are the basics. Again, apply, utilize an amino acid table, while pairing the start and stops and the protein. We just covered that. This is simple. Okay. And the keys here is that this is just basic cell biology. It builds whatever mRNA comes out. The keys here I'm trying to point at is that we're starting to think about how could, for genetic diseases, how could we change the mRNA that comes out? And where are the points that it can be changed? Okay, final thing. I'm going to pull up what we're going to work on right now, okay? Get a new window. Okay. Let's get back to Blackboard, and I want to show you guys a couple things. Number one, since you're all on your computer already, I would like you to pull up. Again, at 3:30 we'll start this. It won't take terribly long. Go here. Again, I posted up here. This is the right recitation link. Apparently the previous one that I linked you was only the Thursday link, and then it was very confusing. So click there. This is our actual Zoom meeting link. Okay. Go here. I'll show you this, okay? I'm not going to show you the exam, obviously. But here's the information about how to access the exam. It's going to be available tomorrow, all right? But here it is. It'll pop up in this online exams in your main course content, okay? You must do this first. That's the online exam honor code. The key here is that I'm telling you can't use outside sources that are not part of class. Okay? No looking stuff up on the internet. And no No like typing it into Yahoo Answers and such. The idea is that this is an honor code. I cannot enforce this heavily. I'm obviously not looking over your shoulder while you're taking it. Neither is the testing center. But please limit what you use to all of the stuff that is posted on Blackboard and your book and your own notes. Okay? You have to fill that out first. Submit either a text submission that says you agree with the honor code or the actual honor code with your name typed in. Once you have that submission, exam 2 will open up to you. You may take it anytime. You must sit down, though, for an hour and a half and take it in one go. It is not available after that. The questions are timed overall. You have as much time as you want to answer any one question. There are 31 questions in total. Okay? Some of them are bigger, some of them are smaller. A lot of them are worth about 3 points each, and the rest are going to be worth between 4 and 8 points depending on the difficulty. Okay? You have access to the entire test at once. I suggest you go through it first and make sure you understand what's where. And I have created basically about 3 different tests, and they're going to be all jumbled together. So if you take it together with someone else, they may have a completely different test than you. Particularly, if you live in the same house, a couple of you guys do live in the same house, okay, you will both have different exams that you're taking, okay? Again, all the questions are presented all at once. You may look at the entire exam from the get-go. Go back to, change, fix, not one at a time. That's crazy. Don't worry. All right. Hour and a half. The instructions are outlined. You must do the honor code first. And yeah, that's mostly it. Okay. All right. I'm going to run downstairs and get something to drink real quick. And I will be back. Oh, hold on. One more thing. Of course. Here we go. Okay. Go here. It is now available. This is the case study we're going through today. I would like you guys to pull up the student handout, and you can look at the cards. We're not doing the card activity. It's more hands-on than we need. But the student handout is actually very helpful, and we're going to go through this bio-interactive together. You guys can go through it on your own as well. I will record the Zoom session, and you can do it on your own or just watch me go through it, okay? The goal here, again, is to discuss how could we utilize all these varying gene expression pieces and put them all together to make genetic medicine using the central dogma. All right. It is up until midnight Saturday. $11:59. I think it's available for like a few minutes past that. The due date is midnight Saturday, like 11:59 p.m. Saturday. But the actual availability goes for like a half hour past that. So if you don't quite get access to it or you don't quite finish it by that time, you will still have a little bit of buffer. Okay, maybe it's a couple hours after. If you're finishing it at like 2 a.m., not a big deal. But if you try to start it at 2 a.m. on Sunday, you're not going to get in. Okay, great. I'll see you guys in a few minutes, okay? Zoom session link. Put it in here for anyone who didn't see this already, okay? I'm just going to pull up my Zoom. Plop that in there. All right, off to get drinks. Going to end my